Hello everyone. In our today's lecture, we are going to start with the next point from the topic is the cells of acquired immunity. In our previous video, you have seen that the cells of acquired immunity, which are formed from the hematopoietic stem cells, and these stem cells are forming myeloid cells and lymphoid cells. Okay, myeloid cells are responsible for formation of RBCs, WBCs. Okay, while the lymphoid cells are responsible for the formation of B and T lymphocytes. Okay, so the next point from this topic is the cells of acquired immunity. Cells of acquired immunity. Acquired immunity. So there are majorly the lymphocyte cells. There are two cells. The first one is a lymphocytes, and the second one is antigen presenting cells. Antigen presenting cells. These are the major two cells which are especially the cells of acquired immunity. The first one is a lymphocyte, another one is a antigen presenting cells. Further on, these lymphocytes are grouped into two. These are into two. The first one is a T lymphocyte. T lymphocyte. And the next one is a B lymphocyte. Okay. In our previous video, we have studied that there are two types of immunity which is shown by human. That is, one is a cell-mediated immunity, and the another one is a humoral immunity, right? So, these T lymphocytes are responsible for. These are showing cell-mediated immunity. Cell-mediated immunity. Called as CM, cell-mediated acquired immunity. Cell-mediated acquired immunity. While the another one is the humoral immunity. It is simply a humoral immunity, immune system. Okay, cell-mediated immune system. Then, just a second. Cell-mediated immune system. And the another one is the humoral immunity. This humoral immunity means what? Humoral means the antibody production. Okay. In humoral immunity, especially the antibodies are produced, and it is called as especially antibody mediated immune system. Antibody mediated immune system. Antibody mediated immune system. Okay, cell mediated immune system. So focus on these two. The long form can be asked. The cell mediated immune system is shown by T lymphocyte, while antibody mediated immune system is shown by the B lymphocyte. That means our acquired immunity try to defeat the antigen by producing antibodies against that antigen, as well as the another one is by just phagocyting that antigen, they try to defeat that antigen, right? So these are the two main types of immunity generally shown by our lymphocyte cells. So there are two different cells, lymphocyte T and B lymphocyte. Why they are named as a T lymphocyte? Because this T lymphocyte cells, and the another one is the B lymphocyte cells. Listen carefully. These T-lymphocytes cells, as these lymphocytes are formed, all the cells, the lymphocyte as well, these lymphocytes are formed in a bone marrow. Okay, these are formed in a bone marrow. So, formed in a bone marrow. Okay, B lymphocyte are also formed in a bone marrow. Red bone marrow, especially in our red bone marrow. Okay. As they are formed in a bone marrow, but here these are formed in a bone marrow, and for maturation it goes to the thymus. For maturation it goes to the thymus as this lymphocyte which goes to the thymus and get matured in a thymus, so named as a T lymphocyte. So they are named as a T lymphocytes. Why the lymphocytes are called as a T lymphocyte? Those lymphocytes which are formed in a bone marrow get matured in a thymus, okay, and then these are called as a T lymphocyte. Okay, as you know, the lifespan of the lymphocyte is of few days to some months or years also. Okay, so few days life cycle is especially is of a B lymphocytes. Okay, and many years, 4 to 5 years lifespan is of a T lymphocyte. The cells which get formed in a bone marrow but mature in a thymus called as a T lymphocytes, and its lifespan is about 4 to 5 years. 4 to 5 years is their lifespan. Okay. While B lymphocyte cells which get formed in a bone marrow get matured in a bone marrow itself. Okay, especially in case of mature in a bursa. Okay, bursa fabricus. Okay, formed in a bone marrow or the bursa fabricus. Okay, as these are formed in this bone marrow and get mature in this, so these are named as a B lymphocyte. As named as a B lymphocytes, and their lifespan is few as compared with the T lymphocyte. Clear, everyone? Now, further on, this T lymphocyte are showing cell-mediated immune system or immune response, while B lymphocyte shows antibody mediated immune system. So you can differentiate B and T lymphocyte here. The short note can be asked, differentiate between the B and T lymphocyte. Okay, the B lymphocyte both are formed in a bone marrow. Okay, mature in a thymus, mature in a bone marrow itself, or bursa fabricus. Okay. So named as a T lymphocyte, so named as a B lymphocyte respectively. Then its lifespan is about 4 to 5 years, and its lifespan is of few days. Okay, and these are showing cell-mediated immune response or immune system, while the B lymphocyte are showing the antibodies-mediated immune system. Clear, everyone? Now we are turning towards the next point, that is the T lymphocyte. So we are dealing with a T lymphocyte. These T lymphocytes are playing an important role in a cell-mediated immune system. Okay. So this cell-mediated immune system, how this works? When antigen invades to our body, okay. When antigen invades to our body, it stimulates our lymphocytes, especially the T lymphocyte. Here I am mentioning the T lymphocyte. When antigenic invasion occurs or antigen enters in our body, at that time our T lymphocytes get sensitized. These cells get sensitized. Okay. On sensitization, these T lymphocyte cells are forming their clones. They are forming their clones. That means the number of T lymphocytes get multiplied. Okay, it means forming the clones means the number of T lymphocytes get multiplied. Now, as the antigen invades in our body, our T lymphocyte gets sensitized and they multiply. They forms their multiple copies. So now we lymphocytes. But this T lymphocyte, the clone lymphocytes, are same but performing different function. Okay, as now we have number of clones, but these clones are performing different function. These are performing different function, and according to their function wise, they are grouped into 4. These are grouped into 4. So now we are here just the types of T lymphocytes. So now we are turning towards the T lymphocytes. Clear, everyone? How? Just when antigen invades in our body, our T lymphocyte gets sensitized. Okay, and they forms their own clones. That means they multiplied in number, and these multiplied or clone lymphocytes are structurally same but performing different function, and because of they are performing different functions, they are divided into 4 types. Okay, the first one is a helper T cells, T lymphocytes, and their clones are the first one is a helper, killer, suppressor, and memory. These are the 4. Okay, so helper T cell, killer T cell, suppressor T cell, and memory T cell. Clear, everyone? So on sensitization, the lymphocyte multiplied and they perform different function, and according to their function, they are derived into 4 or divided into 4: helper, killer, suppressor, memory. Remember, helper, killer, suppressor, and memory T cells. These are the 4 different types of T cells. Okay, T lymphocytes. So related with the helpers. Helper T cells perform so many functions. They are performing so many functions, especially the first function performed by the helper. As the name helper, okay, as the name helper, it performs so many functions. It performs so many functions. Okay, many functions are performed by the helper T cells. These helper T cells secretes lymphokines. It secretes lymphokines. And this lymphokines performs various functions. Okay, these lymphokines perform various functions. That means the first one, it stimulates another T cells. It stimulates another T cells to multiply, right? It stimulates the T cells, so the multiple copies of T lymphocyte get formed. Okay, and the another one, it also stimulates. It stimulates our B lymphocytes. It stimulates our B lymphocytes. So what happened? What is the function of these B lymphocytes? It stimulates our B lymphocytes. So in a result, these B lymphocyte produces the antibodies. It produces the antibodies. Listen carefully. Helper, as the name helper T cell, these helper T cells perform so many functions. One of the major function of the helper T cell is the secretion of lymphokines. This lymphokine secreted by the T lymphocyte when antigen invades through it, at that time the lymphokines, which is secreted by the T lymphocyte, which stimulates the another T cell, and so that the another T cell forms their multiple copies, right? Then the next one, the another function of lymphokine, this lymphokine also stimulates our B lymphocyte. As you know, the B lymphocytes are responsible for antibody mediated immune system. Okay, that means they are producing number of antibodies now. We have to defeat that antigen either by phagocyting it or by producing antibodies against it, right? So this is all related with the helper T cell. Clear, everyone? Its name itself indicating it helps another T cell to multiply and B cells to form antibodies. Clear, everyone? Now the next one is a killer T cell. So related with the killer T cell, this killer T cell, name itself indicates that it is also called as a natural killer cells. It is also called as a natural killer cells. So, natural killer cells. Okay, NK cells. Named as the NK cells. Okay, so related with this NK cells, they are secreting, responsible for cell-mediated immune system and responsible for secretion of perforins. It secretes perforins. Yes. Secretes perforins. So how does natural killer cells work? Listen. Okay. Suppose this is the antigen. This is the any antigen, maybe bacteria, virus, which invades in our body. So what happens? These killer cells get attached. NK cells get attached to. This is a killer T cell. Killer T cell. Okay. When antigen comes, okay, then what happens? This killer T cells goes and binds to that antigen. Okay, and it releases the perforin. What is released by them? It releases the perforin. This released perforin makes whole. Okay, it makes whole, and this is the nucleus, right, of this antigen. Okay, and this perforin works on it and destroy this cell, and thus it destroys the cell. So it is called as a natural killer cell. So it is called as a natural killer cell because it releases the perforin and this perforin kills that cell. Okay, as generally the NK cells, natural killer cells, perform so many functions in our body, especially they remove the carcinogenic cells in our body. Okay, especially it works on the antigen also, as well as it also releases the destroys the cancerous growth in our body. So these are the natural killer cells releasing the perforins. Then the another one is a suppressor. Okay, sometimes suppose some cells, when the antigen invades in our body, at that time some cells invade this and engulf this antigen inside. Okay. So now by forming a phagocytic vesicle, suppose this is our macrophage cell, and this macrophage cell has taken the antigen inside. Okay, that means it is forming a phagosome inside. So what happened? When this natural killer cells it bind with this cell, it releases the perforin and also kills this. Clear? Likewise, the natural killer cells works. But sometimes what? So what is the role of the suppressor cell I'm talking about? The suppressor cells stops the autoimmune response, especially works. Sorry, it prohibits the bad effect of this perforin on our own cells. So thus it protects our own cells. Clear, everyone? These cells tries to protect the suppressor T cells, nullifies the effect of killer cells on our own cells. Clear? And protects our own cells. This is about the suppressor T cell. As the name, it suppresses the immune response which is shown by the killer T cells, and thus it protects our own cells. Then the next one is the memory T cells. As the name memory means what? It keeps it in mind that previously that antigen invaded us. That means previous antigenic invasion is kept into mind. That is done by the memory T cell. And when the next time when that antigen tries to invade us, at that time these memory T cells recognize that antigen and tries to destroy it. So this is a very important function, that is a memory T cell, which keeps the previous attack in a memory. Okay, so this is all related with the T lymphocyte. Once again, just revise the T lymphocyte. The T lymphocyte, which are formed in a bone marrow, get matured in the thymus, so named as a T lymphocyte. Okay, when antigen invades to the body, at that time the T lymphocyte gets sensitized. The sensitized T lymphocyte forms their clones, that means forms their multiple copies, and these multiple copies cells which are performing different function, and according to their function they are grouped into 4: helper, killer, suppressor, and a memory T cell. Clear? Okay, so the helper, as the name helper, it helps or it performs so many functions. Among that, it secretes, the first one is the lymphokines. This lymphokine stimulates the T lymphocytes to form their multiple copies, as well as it also stimulates the B lymphocyte to produce antibodies. Then then the killer T cells. The killer T cells are also called natural killer T cells. These cells are responsible for secretion of perforins, and this perforin forms the hole in a cell and destroy the antigen. Okay, then suppressor, it suppresses the immune response, especially which is shown by the killer T cell, and protects our own body cells. Then memory T cells are responsible for, especially this keeps the previous memory in a mind. Okay, so this is all related with the cells. The helper T cell is also called as the another names. Okay, remember these are called as the CD4 cells. Helper T cells are called as a CD4 cells, or it is also called as a T4 cell. Helper is called as a CD4 or T4 cells. Then the next one is a killer T cell, is called as CD8 cell. CD8 cell, or it is also called as a T8 cell. Clear, everyone? Then the next one is a suppressor, and a memory doesn't having another name. Clear, everyone? So helper is a CD4 or T4 cell, while killer is called as a CD8 or T8 cell. Clear? So this is about lymphocytes. Now we are turning towards the B lymphocyte. Okay, so related with the B lymphocytes are grouped into 2 only. Okay, the first one is the plasma cell and memory B cell. Okay, it is grouped into 2. The first one is a plasma B cell, and the another one is the memory B cell. As you know, this B lymphocyte are responsible for antibodies mediated immune system. immune system, right? AMIS. Okay. So related with the plasma B cells. So these plasma B cells are known to be a factories for antibodies production. These are known to be a factories for antibodies production. Antibodies production. Near about 2,000 molecules or antibodies are produced per second. That means its production rate is more numerous. Okay, near about 2,000 molecules antibodies are produced per second by the plasma B cells. This plasma B cells plays a very important role in a humoral immunity by producing the antibodies, and it is very specific to that antigenic invasion. Then the next one is a memory B cell. The memory B cell, as the function of memory T cell, the just same function is performed by the memory B cell. It keeps in mind the previous attack or invasion of that antigen. So this is all about the types of lymphocyte, B and a T lymphocyte. Clear? Okay. Now the another one, lymphocyte, and the another one is the antigen presenting cell. Here we complete only the lymphocyte. Now the another cell is the antigen presenting cell. So first point was the lymphocyte, and the second one is a antigen presenting cells. So the related with the antigen presenting cells. Okay. Generally the helper T cells. Here are the helper T cells. Perform the function. Okay, these are the antigen presenting cells. So just which cells are called as the antigen presenting cells? So especially our just a second, our macrophage. Macrophage cells, then dendritic cells, and even though the B lymphocytes. These cells are called as the antigen presenting cells. These cells are called as the antigen presenting cells. So how the antigen presenting cells works? Clear. So when any antigen invades in our body, to our body, at that time the macrophage shows cell mediated immunity, right? They goes to that antigen. Suppose this is our macrophage cell. This macrophage cell forms by phagocytosis. This is taken inside. This antigen is taken inside. At that time, in a macrophage, the lysosomes, which releases the lytic enzymes, and try to digest this antigen into small fragments, forming this is the lysosome, which is present in a macrophage, which having lytic enzyme. Because of the action of this lytic enzyme, the antigen get broken into the small fragments. These are the fragments. Okay, and there are some proteins to which this fragment get bind, and then it forms It comes on a surface, this protein, and to that protein, here are some proteins which are present in the macrophage cell. To this fragment get bind. Which is of that? This fragment is of a antigen. Clear? So this is the fragment and this is the protein to which the fragment of this antigen get bind, which forms a flag-like structure to this antigen, to this cell, macrophage cell. Now it doesn't having the antigen now. Okay, that means this macrophage cell, which having previously it having the antigen, but the antigen was digested or broken down into the small fragments, and this fragment get bind to the protein and comes on a surface. Clear? So those cells which has digested such an antigen or broken such an antigen, okay, these are called as the antigen presenting cells. Clear, everyone? That means some cells, our body cells, which are especially doing a function of phagocytosis, they phagocyte that antigen, digest it, broken down into the small fragments, and these fragments, okay, comes on their surface. Now these cells are called as the antigen presenting cells. Okay, clear, everyone? Now these are the antigen presenting cells. Then what happened? Now this antigen presenting cell which having the antigen, okay, digested the antigen. So this antigen presenting cell stimulates our helper T cell. Okay, this antigen presenting cell. Now it is not a macrophage only. It is an antigen presenting cell. Funda clear? Okay, so antigen presenting cell. Clear, everyone? This antigen presenting cell stimulates our T lymphocytes. It stimulates our T lymphocyte. Okay, especially the helper T cell. This helper T cell, it stimulates our helper T cell. Helper T lymphocytes. Stimulates our helper T lymphocyte. So as the helper T lymphocyte gets stimulated, it stimulates B lymphocyte. B lymphocyte. And you know the function of B lymphocyte. The B lymphocyte produces the antibodies. So our body destroy the antigen by phagocyting it, as well as by producing the antibodies against it. Yeah. So thus the antibodies are also produced to destroy that antigen. Okay, antigenic invasion is in a large amount. Okay, so we have to defeat that antigen by two ways: by phagocyting it, as well as by producing the antibodies. So the antigen presenting cells also destroy tries to destroy the antigen by stimulating our lymphocytes, B lymphocyte, by producing the antibodies. Clear, everyone? So there are majorly the macrophage, dendritic cells, and B lymphocytes work as an antigen presenting cell. And how it works, especially our macrophage cells phagocyte that antigen, destroy that antigen inside, mix it into the small fragments. Then these fragments along with the protein comes on a surface and bind to the surface. Now this macrophage is turning as a antigen presenting cell, and which is recognized by or it sensitizes the helper T cells or stimulates the helper T cell, and helper T cell, as its function, it stimulates the B lymphocyte, and the B lymphocyte produces the antibodies. So this is whole about the cells of acquired immunity, lymphocytes and antigen presenting cells. Lymphocytes are categorized into two. The first one is a T lymphocyte and the B lymphocyte. T lymphocytes are of four types: helper, killer, suppressor, and memory T cells, while B lymphocytes are of two types: plasma B cell and a memory B cell. Plasma B cells are known to be factories for antibodies production, while the memory B cell is keeping the memory of the previous attack. Okay, and another one is the antigen presenting cell. Okay, so this is all related with the cells of acquired immunity. Now we are turning towards the next point of the topic, that is the structure of antibody. So related with the structure of antibodies. This question can be asked, okay, very important point. The structure of antibody can be asked for 2 or 3 marks question. So related with the antibodies, antibodies are also called as immunoglobulins. Immunoglobulins. Okay. Abbreviated as IG. Okay. Antibodies. When antigenic invasion occurs to our body produces specific type of proteins. Okay, as these are the proteins. Globulins means proteins. Okay, globulins are the proteins which participate in an immune response, so named as a immunoglobulins IG. Okay, majorly the antibodies are Y-shaped. Majority the antibodies are Y-shaped. Antibodies are Y-shaped antibodies. Majorly these are Y-shaped. Okay. These immunoglobulins are produced by, or antibodies are produced by, yes, B lymphocytes. More precise, B cells of a B lymphocyte, especially the plasma B cells. Plasma B cells, okay, are responsible for the production of antibodies. These are responsible for the production of antibodies. So, numerous antibodies are produced by the per second, near about 2,000 molecules are produced per second, and so these plasma B cells are known to be factories for antibodies production. So majority these are Y-shaped. These antibodies are made up of 4 polypeptide chains. Antibodies are made up of 4 polypeptide chain. Chains. Among these, 2 are the heavy chain. 2 are the heavy chain, and 2 are the light chain. 2 are the light chains. Heavy as well as light chain. So just see the structure of the antibody. So majority of the antibodies are Y-shaped. So the question can be asked for sketch and label. So you can just see the diagram how I am drawing, and just you have to draw the diagram in your notebook also. So this is a Y-shaped. Okay. So this is the Y-shaped structure. Okay. Just to its outer side. Here is the another one. So what I have drawn with the white chalk, that is the heavy chains. These are the heavy chains. And with the yellow chalk, these are the light chains. So these are the light chains. So majorly this is the structure. So These are the heavy chain. Heavy chain. And this is the light chain. Clear, everyone? Now, both this, suppose this shaded region is called as a, just a second. Up to this, the shaded region is called as a constant region of the heavy chain. So this is the this shaded region is called as a constant region. Constant region. And this unshaded part is called as a variable region. Variable region of a heavy chain. Okay, H for heavy chain. Okay. Then the light chain also having constant region. So this is the shaded region of the light chain. Here I am just shading the parts. So this is the constant region of a light chain. Of a light, L for light chain. Both and this is the variable region of a constant as well as heavy chain. It having, here is the antigen binding site. Here is a site called as a antigen binding site. Okay. So this is named as a antigen binding site. This antigen binding site which is present on the antibody is called as a paratope. It is called as a paratope. Okay. Okay. These heavy chains are binded with each other by disulfide bond. Here is the disulfide bond to which these are the disulfide bond. Both the heavy as well as light chains are also binded with each other by a disulfide bond. Here is also a disulfide bond. So clear? It having two terminals. One is the C terminal, both light as well as heavy chain, and the another one is the N terminal. One is the carboxyl end and one is the amino terminal. Okay. So this is the structure of antibody. This is the region called as a hinge region. Slightly fluctuating region, hinge. Slightly movable region. Okay, so listen carefully. The structure of antibody, how we have to write. So, majority of the antibodies are also called as immunoglobulins. These are also called as Ig, abbreviated as Ig. These antibodies are produced by the plasma B cells. The plasma B cells are the factories for antibodies production. Majority of the antibodies are Y-shaped. Majority of the antibodies are Y-shaped. These are made up of 4 polypeptide chain. These are made up of 4 polypeptide chain. Okay, this is the one which is big one. These are the big one. These are called as a heavy chain. Okay, listen. It consists of 2 heavy chain and 2 light chain. Right? The heavy chains, why it is named as heavy chain? Because of these are the polypeptide chain. Clear? These polypeptide, the heavy chains are made up of 440 amino acids. The number of amino acids present in a heavy chain is more, so it is called as a heavy chain. How many amino acids are there in a heavy chain? 440 amino acids. While in a light chain, they are having 214 amino acids. 214 amino acids. Okay, so clear everyone. Majority of the antibodies are Y-shaped. It consists of 4 polypeptide chain. Among these, the 2 are the heavy chain, 2 are the light chain. The heavy chain are made of 440 amino acid, and the light chain are made up of 214 amino acid. Clear everyone? Both the heavy chain as well as light chain having constant region. This is the constant region of light chain, and this is the constant region of a heavy chain. Both the heavy as well as light chain having the variable region. Clear everyone? First sentence, it is also called as immunoglobulins. Antibodies are also called as immunoglobulins. These are produced by the plasma cells. Okay. Majority, the antibodies are Y-shaped. It consists of 4 polypeptide chain. Among these, the 2 are the heavy chain and 2 are the light chain. The heavy chains are made up of 440 amino acids, and the light chains are made up of 214 amino acids. Clear? Both the heavy as well as light chain having the constant region as well as the variable region. Clear everyone? Okay, the constant region of both the heavy as well as light chain having C terminal or carboxyl end, and the variable region having amino terminal or N terminal. Clear everyone? Okay, then at the variable region of both the constant heavy chain as well as light chain having antigen binding site. So this antigen binding site at where the antigen has to bind, this site is called as a paratope. This site is called as a paratope. Clear? Okay, this site is called as a paratope. Now, both the heavy chain are binded with each other by disulfide bond. Yes, it is binded with each other by disulfide bond. As well as the heavy as well as the light chain are also binded with each other by the disulfide bond. Okay, and there is a hinge region. Okay, here is the hinge region, and where there is also a disulfide bond. Okay, so this is all related with this. There are near about 16 disulfide bonds are present in a antibody. Clear everyone? So this is all related with the antibody. So one more point I will explain. The heavy chain having 440 amino acids. So the heavy chain having a variable region. This variable region, heavy chain having a variable region, which is made up of near about 118 amino acids. How many? 118 amino acids. So the constant region of a heavy chain, is made up of 1192. Yes, how many? 440 amino acids. Clear? So this is the big one. Clear? The constant region is a big one. Then the next one related with the light chain. Light chain also having a variable region. This variable region of a light chain is made up of near about 108 amino acids. Okay, 108 amino acids. While the constant region of the light chain, it is made up of 1092. Yes, how many? 214 amino acids. Okay, clear everyone? So the constant region, clear? Listen here. The light chain is made up of 214 amino acids totally. The variable region of the light chain is made up of 108 amino acids, and the constant region is made up of 109 to 214 amino acids. Clear to everyone? This is and this one. Now related with the heavy region, heavy chain. Heavy chain is made up of 440 amino acids. Among this is the variable region. This variable region is made up of 118 amino acids, and the remaining is made up of 119 to 440. Clear to everyone? So this is very simple one. So just revise the structure of antibody. Take a glance on it. Antibodies are also called as immunoglobulins. These immunoglobulins are majorly the Y-shape produced by the plasma B cells, known to be factories for antibodies. Okay, these are made up of 4 polypeptide chain. Among these, the 2 are the heavy chain. The heavy chain are made up of 440 amino acids, and the light chain are made up of the 214 amino acids. Both the heavy as well as light chain having constant region, okay, and the variable region. The variable region is called as antigen. At the variable region of both the heavy as well as light chain having antigen binding site to which the antigen has to bind. Clear? Both the heavy as well as both the heavy chain are binded with each other by disulfide bond, as well as both the heavy as well as light chain are binded with each other by disulfide bond. Clear? It having at the constant region of both the heavy as well as light chain, it having C terminal or carboxyl end, and at the variable region of both the heavy as well as light chain having N terminal, that means amino terminal. So this is all related with the structure of antibody. So here we will stop now. Thank you.